Hello everyone, this is the first lecture in our presentation on gluconeogenesis, glycogen metabolism and the pentose phosphate pathway. In this section we want to answer the questions: what is the nature of gluconeogenesis, the pathway that synthesizes glucose from non-carbohydrate precursors? How is glycogen synthesized from glucose and hydrolyzed when it is required? And how are electrons from glucose used in biosynthesis? So just as a quick overview of glucose metabolism at this stage, what we've looked at during our lectures on glycolysis is we've seen how glucose, when it enters the cell, gets phosphorylated to glucose 6-phosphate, and then the rest of the glycolysis reactions turns that into pyruvate, and of course pyruvate can be decarboxylated to acetyl coenzyme A and carry on into the citric acid cycle. Under anaerobic conditions in mammalian cells, pyruvate will be turned into lactate, or it can also be a precursor for production of amino acids. What we will look at now is a biosynthetic pathway where pyruvate and other precursors can be used through a process called gluconeogenesis to form glucose 6-phosphate or even glucose, and that can be used in the body where it is required. It can, for example, be used to produce glycogen, which is a form which mammalian bodies use, a polymer which mammalian bodies use to store glucose for when energy is required. Or it can be shunted towards the pentose phosphate pathway to produce the 5-carbon sugars which is required for things like RNA. Or, of course, during that pathway you will see we also can produce NADPH, which is the coenzyme most used to produce reducing power for biosynthetic pathways. So we are shifting then from catabolism to anabolism, and just briefly a few principles of biosynthesis. Often anabolic and catabolic pathways use the same reversible reactions. We spoke about amphibolic reactions being reactions that can serve both catabolism and anabolism. But of course, when we talk about pathways, at least one of those reactions in the pathway has to differ. Anabolic and catabolic throughput is then defined by the metabolic needs. Anabolic and catabolic pathways are controlled by one or more reactions unique to that specific pathway, normally at an early step in the pathway so that we don't waste energy and metabolites. And what we say is that this control has to be reciprocal, which means that when anabolism is switched on, catabolism must be switched off. Now, we did say in an earlier lecture that anabolism and catabolism are not mutually exclusive, which means that, for example, glycolysis, a catabolic pathway, can take place at the same time that certain amino acid synthesis pathways can take place, which are anabolic processes. But when we talk about amphibolic pathways, so pathways which will use some of the same enzymes to do either a catabolic process or an anabolic process, we cannot have both of those processes switched on at the same time. So biosynthetic processes are often coupled to ATP hydrolysis so that the overall process is irreversible in vivo when required and favorable even when reactant concentration is low. So then on to gluconeogenesis. It is the generation of new glucose from common metabolites. Human metabolism requires about 160 grams of glucose per day, and about 75% of that is used by the brain alone. Body fluids normally contain about 20 grams of dissolved glucose, and you have a further up to 200 grams of glucose stored in the form of glycogen, which is a polymer of glucose molecules. Glycogen then stores are partially depleted during strenuous exercise, and for these reasons the body must be able to make its own glucose. It has to be able to restore the amounts of stored glycogen, and it has to be able to sustain normal activity through reforming the minimum blood glucose level that is required, etc. So the major sites where gluconeogenesis takes place is in the liver cells, which accounts for about 90% of gluconeogenesis, and about 10% of gluconeogenesis takes place in kidney cells. And the glucose produced in liver and kidneys are then released into the blood and transported to the brain, the heart muscles, and red blood cells where it might be required. So gluconeogenesis then is the process whereby precursors such as lactate, pyruvate, glycerol, and amino acids are converted to glucose. Most of those precursors enters the TCA cycle at some point and are converted to oxaloacetate, and we'll see oxaloacetate is a precursor that can be used in this pathway in order to produce glucose. So gluconeogenesis then is not just the reverse of glycolysis. Glycolysis is an exergonic reaction, which means that the overall process has a negative delta G of negative 74 kilojoules per mole, which means that the reverse of the process is going to, by using the same reactions, is going to be a positive 74 kilojoules per mole, which means that it won't be a thermodynamically favorable process and it won't take place spontaneously. So if gluconeogenesis were just the reverse of glycolysis, its delta G would be positive and it wouldn't happen spontaneously. So the energetics therefore have to change in order to make gluconeogenesis favorable as well. And the regulation of these two processes are of course also necessary so that when glycolysis is active, gluconeogenesis is inhibited and vice versa. So how do we change the energetics of the process and allow regulation of both of these processes? Well, of course, there are certain reactions which will be common to both glycolysis and gluconeogenesis, but we will have to require some new enzymatic reactions in order to bypass some of the glycolysis steps. So gluconeogenesis will basically be something old, something new, something borrowed, nothing blue, unfortunately. But you will recall that 7 of the 10 glycolytic reactions were close to equilibrium, which means they are reversible. So under cellular conditions, the delta G's for those reactions, reaction number 2 and the reaction number 4 to 9, we saw were all fairly close to 0. But for hexokinase, phosphofruktokinase, and pyruvate kinase, we have relatively large negative delta G's. And that means these reactions must be bypassed because they are irreversible. The new reactions provide for a spontaneous pathway with a negative delta G in the direction of sugar synthesis, and they also provide new means of regulation. So by bypassing these reactions with new reactions, we will see overall a pathway which can be spontaneous in the direction of sugar synthesis, in other words in the gluconeogenesis direction, and of course those new reactions are also amenable to regulation. Okay, so to take a broad view of glycolysis and gluconeogenesis stacked up against each other, you are now well aware of the reactions of glycolysis in this forward direction, taking glucose onto pyruvate, and we will see that the 3 steps that we spoke about on the previous slide that have to be bypassed is turning pyruvate back into phosphoenol pyruvate, which actually happens in a 2-step process using pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Then there is a fructose 1,6-bisphosphatase, which is a dephosphatase reaction to turn fructose 1,6-bisphosphate into fructose 6-phosphate, bypassing the PFK phosphofruktokinase reaction of glycolysis. And then finally, to bypass the hexokinase reaction, we use an enzyme called glucose 6-phosphatase, and again it is a dephosphorylation that happens at that reaction. Okay, so into the details of these bypasses then. The first bypass, the pyruvate kinase bypass, the last step of glycolysis is catalyzed by pyruvate kinase, and it turns phosphoenol pyruvate into pyruvate, and it does a substrate level phosphorylation in the process to produce ATP. So for the bypass of pyruvate kinase, we require the cleavage of 2 phosphate bonds, and the cleavage of 1 phosphate bond is a usage of an ATP actually is not sufficient energy to drive the synthesis of phosphoenol pyruvate. So simply using 1 ATP does not give us enough energy to turn pyruvate into phosphoenol pyruvate, and this is why this happens in a 2-step process, and we invest 2 ATP equivalents. So the bypass of the pyruvate kinase therefore requires two enzymes. It requires pyruvate carboxylase, which uses a pyruvate and bicarbonate and an ATP to produce oxaloacetate, and an ADP and inorganic phosphate is released. So this is this reaction over here. A bicarbonate and an ATP is used to produce oxaloacetate. So carbon is actually added to this pyruvate structure in order to form oxaloacetate. And then the second part of the reaction uses a phosphoenol pyruvate carboxykinase, or PEPCK, enzyme and a GTP to turn oxaloacetate into phosphoenol pyruvate, and it releases a GDP and it releases a carbon dioxide as well. In fact, it's this same carbon dioxide that was added to pyruvate that now is decarboxylated to form phosphoenol pyruvate. So the energy then that contributes to the spontaneity of this reaction actually comes from three things. We have free energy from one phosphate in the form of an ATP that is utilized. We have a spontaneous decarboxylation reaction that happens between oxaloacetate and phosphoenol pyruvate, and of course the cleavage of second phosphate, this time in the form of GTP, that is used in the PEPCK reaction. So the pyruvate carboxylase, the first of those two reactions, uses a biotin prosthetic group. The pyruvate carboxylase reaction is the initiation of the gluconeogenesis pathway, and carboxylations that use bicarbonate as a substrate, as a carbon source, always use biotin as a coenzyme. We'll see this in our Biochemistry II course when we talk about fatty acid synthesis as well, that those enzymes also use biotin as coenzymes. So the reaction requires ATP and a bicarbonate as substrates, and the biotin is covalently linked in the enzyme's active site, specifically on a lysine amino acid residue. Acetyl coenzyme A is an allosteric activator of this enzyme, and its regulation works as such: when ATP or acetyl coenzyme A are high, pyruvate enters gluconeogenesis. So if you think about it, it kind of makes sense. If ATP is high, it means the cell has a high energy state. If acetyl coenzyme A is high, it means there's already a high carbon flux, which can either be used in the TCA cycle to produce more energy or other precursors, or it can itself be used as a precursor to enter gluconeogenesis. So the cell is rich both in energy and in carbon, and it can save that carbon in the form of glucose, and therefore the cell can enter gluconeogenesis. The second part of bypass 1 is the PEP carboxykinase, or PEPCK enzyme, that catalyzes the conversion of oxaloacetate to phosphoenol pyruvate. So the PEPCK uses GTP and a decarboxylation to drive the synthesis in the forward direction. So lots of energy is needed to drive this reaction forward, and it's provided in both of those ways, both by the spontaneous decarboxylation, which is a favorable reaction. So this carbon dioxide that was added in the previous step is now decarboxylated in this reaction, and a GTP is hydrolyzed, and it's equivalent to ATP in this reaction. So as I said, the carbon dioxide that was added to the pyruvate in the pyruvate carboxylase reaction is the one that is removed now in the PEP carboxykinase reaction, and we end with phosphoenol pyruvate. So that brings us now to the second bypass. So from phosphoenol pyruvate all the way up to fructose 1,6-bisphosphate, all of the reactions that were used in glycolysis are now used in the reverse direction, because of the regulation that the cell is under that allows the reactions to happen in the synthesis direction, remembering that those of course are reversible reactions with a delta G very close to zero under cellular conditions. So this now brings us to the need of hydrolyzing fructose 1,6-bisphosphate to fructose 6-phosphate. In other words, what we want to do is we want to bypass reaction 3 of glycolysis, catalyzed by phosphofruktokinase. So phosphofruktokinase, you'll recall, catalyzes the reaction fructose 6-phosphate plus ATP to fructose 1,6-bisphosphate and releases an ADP in the process. So this is a coupled reaction where the phosphorylation is coupled with the use of ATP, the dephosphorylation of ATP, which is an hexagonic reaction, and that free energy is used to produce fructose 1,6-bisphosphate. So that means that the reverse reaction, the phosphatase reaction catalyzed by this enzyme fructose 1,6-bisphosphate, can actually happen spontaneously. So fructose 1,6-bisphosphate in water, because it's a hydrolysis reaction, yields fructose 6-phosphate and an inorganic phosphate. So as I said, it's a thermodynamically favorable reaction. The delta G in the liver is approximately minus 8.6. It's also an allosterically regulated enzyme, where citrate will stimulate the enzyme, and fructose 2,6-bisphosphate will inhibit the enzyme, and AMP will also inhibit the enzyme. And in fact, the inhibition of AMP is enhanced by fructose 2,6-bisphosphate. Now, a little later on in the lecture, we will spend more time on fructose 2,6-bisphosphate, but you will recall from our talks on the regulation of glycolysis that this metabolite enhances glycolysis even in conditions where there's a lot of ATP available. At the same time, it actually inhibits the reverse reaction, in other words, this bypass reaction. So it has an effect both on activating glycolysis and inhibiting gluconeogenesis. And it also enhances the inhibition of gluconeogenesis when AMP is already available. It enhances the effect of AMP. Then the final bypass is the bypass of hexokinase, in other words, the first reaction of glycolysis, where glucose is phosphorylated using ATP to form glucose 6-phosphate. So the gluconeogenesis reaction requires glucose 6-phosphatase enzyme to dephosphorylate glucose 6-phosphate and leave us with glucose and inorganic phosphate. So glucose 6-phosphatase enzyme is actually embedded in the endoplasmic reticulum of liver cells, and the catalytic site is exposed to the ER lumen. So another subunit may then actually function as a translocase, providing access to the substrate substrate to the active site. In other words, providing the glucose 6-phosphate to come into contact with the active site of the glucose 6-phosphatase enzyme. And the presence of glucose 6-phosphatase in the ER of liver and kidney cells is what makes the final step of gluconeogenesis possible. Glucose 6-phosphate is hydrolyzed after it's taken up into the ER, and this system includes the phosphatase itself as well as 3 transport proteins. And these transport proteins are just called T1, T2, and T3, and the way that this works is that the other reactions of gluconeogenesis that happens in the cytosol will produce the glucose 6-phosphate. The T1 transporter will then allow glucose 6-phosphate to be transported into the ER lumen. The glucose 6-phosphatase enzyme, which is embedded in that ER membrane, but it has its active site directed towards the ER lumen, will now turn glucose 6-phosphate into glucose and phosphate. And then 2 transporters, T2 and T3, will respectively carry the glucose and the phosphate out of the ER lumen. And a separate transporter called the GLUT2 transporter can then transport glucose out of the liver cells and into the blood cells. And this is what allows the liver to increase the blood glucose levels. Now, the reason that this slightly complicated system is in place is that glucose 6-phosphate actually doesn't always need to be turned into glucose during gluconeogenesis. When the object is to make glycogen, glucose 6-phosphate is actually the one of the precursor molecules to form glycogen. So we don't actually need to form glucose as such, but of course when blood glucose levels are quite low, it is required that that glucose is formed so that it can be excreted by the cells into the blood. So then, just as a summary of what we've just said, this is the gluconeogenesis pathway written over the glycolysis pathway. The glycolysis enzymes I've made in blue on here, and the gluconeogenesis enzymes are in red. So what we've said is that pyruvate is turned into phosphoenol pyruvate in gluconeogenesis by using a 2-enzyme system. Pyruvate carboxylase first turns pyruvate into oxaloacetate, and then PIP carboxykinase turns that into phosphoenol pyruvate. In the process, a bicarbonate, an ATP and a GTP is used, and in the second step, the spontaneous decarboxylation helps to drag this reaction forward. Then the other two bypasses includes the fructose 1,6-bisphosphatase, which bypasses the phosphofruktokinase reaction to dephosphorylate fructose 1,6-bisphosphate to fructose 6-phosphate, and the final reaction to turn glucose 6-phosphate into glucose is catalyzed by glucose 6-phosphatase. Then just briefly, let's discuss the so-called Cori cycle. So the Cori cycle is something that operates during strenuous exercise. For a brief burst of energy, muscle cells will utilize phosphate stored as phosphocreatine. And once phosphocreatine is exhausted, ATP is provided in muscle cells mainly by glycolysis, with the input coming from glycogen, so the sugar released from glycogen, the glucose released from glycogen, and also glucose that is taken up directly from the blood. Now, lactate will be produced during this process from pyruvate. You'll remember that during strenuous exercise, the oxygenation levels are such that oxidative phosphorylation cannot take place at the rate required, and so pyruvate is turned into lactate by lactate dehydrogenase in order for the cell to reform its NAD+ so that glycolysis may continue. So now the cell excretes this lactate into the blood, and this will allow lactate produced from this pyruvate to pass via the blood to the liver, where it can be converted back to glucose. So via the blood, lactate goes into the liver cells. Now the lactate concentration in liver cells are quite high, and lactate dehydrogenase reaction acting in reverse, because lactate concentration is now quite high in these cells, will allow pyruvate to be reformed in liver cells. And then using gluconeogenesis, pyruvate will be turned back into glucose and in the liver. So this glucose formed by gluconeogenesis in the liver can either travel back to the muscle via the blood, or it can be used to synthesize glycogen to store for the next time that strenuous exercise might occur. So this cycling of lactate produced during exercise in the muscles via the blood to the liver to reproduce glucose through gluconeogenesis, which can then again be travel via the blood back to the muscle cells where it is required, is called the Cori cycle. So we look now at regulation of gluconeogenesis. We said that glycolysis and gluconeogenesis are both spontaneous reactions through, of course, a series of coupled reactions that take place in both processes. But if both of these pathways actually run simultaneously inside the cell at the same time, it would constitute a futile cycle that would waste energy. And this is because during glycolysis, glucose is turned into two pyruvate molecules, and a net gain of 2 ATPs take place. But during gluconeogenesis, 2 pyruvates is turned into a glucose, but this costs us 4 ATPs and 2 GTPs. That means because glycolysis yields 2 ATPs or 2 NTPs, and gluconeogenesis actually costs us 6 NTPs in the form of 4 ATPs and 2 GTPs. A futile cycle, if both of the pathways are running at the same time, would waste 4 NTPs per cycle. Now, to prevent the waste of energy on a futile cycle, glycolysis and gluconeogenesis are reciprocally regulated, i.e. when glycolysis is turned on, gluconeogenesis is turned off. So the way that this works is that when the energy status of the cell is high, glycolysis should be off, and pyruvate, etc. should be used for the synthesis and storage of glucose, because the cell already has a large amount of energy available. And of course, conversely, if the cell has a low energy status, glucose should actually be degraded to provide energy, and it shouldn't be built up to store. We differentiate between two measures of control of these processes. The first we call local control, which is the reciprocal allosteric regulation by mostly adenine nucleotides. But we will also talk about global control, which is how in multicellular organisms like mammals, hormonal signals are used to switch certain processes on and off. And we will see that there is an interplay for the regulation of glycolysis and gluconeogenesis through the hormonal control as well. But looking first at local control, the phosphofruktokinase enzyme of glycolysis is inhibited by ATP and stimulated by AMP, as we've mentioned before. And the fructose 1,6-bisphosphate reaction, which is the bypass of that PFK reaction in gluconeogenesis, is conversely inhibited by AMP. Now, when ATP is high, and that means the cellular energy levels is high, the cell should store its energy as glucose, and that is why ATP inhibits phosphofruktokinase and thereby inhibits the glycolysis reaction. But when ATP is low, it will mean that AMP will conversely be high, so the cell does not expend energy on synthesizing glucose. It should actually shut off its gluconeogenesis pathway and turn on its phosphofruktokinase reaction. And this is why we see this reciprocal effect here of AMP. AMP will switch off the phosphofruktokinase the fructose 1,6-bisphosphate enzyme of gluconeogenesis, while also switching on the phosphofruktokinase enzyme, which of course switches on glycolysis. Now, global control in liver cells includes the reciprocal effects of a cyclic AMP cascade, which is triggered by the hormone glucagon when blood glucose is low. So when blood sugar levels are low, glucagon is released, and this will signal to liver cells that a large amount of protein kinases should be switched on through a vast signaling network that we do not need to know the details of. But this cyclic AMP cascade will trigger phosphorylation of enzymes and regular proteins, specifically by a type of kinase which gets activated called protein kinase A. And this results in the inhibition of glycolysis and the stimulation of gluconeogenesis. Because remember now, this is a signal that comes when blood glucose levels are low. So the cell then signals that the glycolysis should come to a halt and gluconeogenesis should happen. Blood glucose levels are low, so we don't want breakdown of more glucose through glycolysis. We actually want production of more glucose by gluconeogenesis. So that'll then make glucose available for release into the blood. The enzymes and proteins relevant to these pathways that are phosphorylated by this protein kinase A, which was switched on by glucagon, include the pyruvate kinase, of course the last reaction of glycolysis. This gets phosphorylated and it is inhibited when it is phosphorylated, so that helps to stop glycolysis. A protein called CREB, or CAMP response element binding protein, is a type of transcription factor which activates through other factors the transcription of the gene pepcarboxykinase, one of the bypass steps in gluconeogenesis, of course, and that leads to the increased level of gluconeogenesis. And then a bifunctional enzyme that makes and degrades an allosteric regulator, fructose 2,6-bisphosphate, is also phosphorylated, and we will see what effect that has. But first we need to talk a little bit more again about this modulator metabolite, fructose 2,6-bisphosphate. We already spent some time on it when we spoke about regulation of glycolysis. We said that fructose 2,6-bisphosphate stimulates glycolysis. It allosterically activates the glycolysis enzyme phosphofruktokinase, that reaction 3, the committed step of glycolysis, and it also allows the activation of transcription of glucokinase in the liver, and that obviously increases the level of glycolysis that can take place in those cells. Now, you'll recall that phosphofruktokinase is the rate-limiting step of glycolysis, the committed step, and it's allosterically inhibited by ATP, and that's what we illustrate in this little image over here. When we have a low level of ATP present, the phosphofruktokinase activity will increase with the amount of its substrate, fructose 6-phosphate, that is present. But when you add ATP into this experiment, we will see that at high ATP concentrations, even though you keep increasing the substrate for the PFK, you actually get much lower activity than you would if ATP was not present, or if it was present at low levels. But This happens because at high concentration ATP binds at a low affinity regulatory site of the PFK enzyme, and that promotes its tense conformation. Now what fructose 2,6-bisphosphate does, it actually gets activated in the presence, it activates phosphofruktokinase because and then allows it to function as it would function if ATP concentration was low. The way that it does this is that it also binds to the PFK enzyme but promotes the relaxed state of the enzyme even at high ATP concentrations. So the activation by fructose 2,6-bisphosphate, whose concentration fluctuates in response to those hormonal signals, will actually then supersede local control by ATP. And what we mean by this is that, so if no ATP or if no fructose 2,6-bisphosphate is present, this is the S-curve that is typical of allosterically regulated enzymes that we see when we plot the relative velocity over increasing substrate concentrations. When we have fructose 2,6-bisphosphate present at various concentrations, the pink line indicates a higher concentration than the green line. We see that because this enzyme is turned into a more relaxed state, even if ATP is present, we will see an increase in the activity of that enzyme. So the second graph shows this with higher ATP concentrations present. So we'll see if we have a high ATP concentration, it pretty much shuts down the velocity of the phosphofruktokinase enzyme quickly. But if we now also have the fructose 2,6-bisphosphate present, we see that much higher ATP concentrations can be tolerated. So this is why we say that the PFK activity in the presence of the globally controlled allosteric regulator fructose 2,6-bisphosphate is similar to that of when ATP concentration was low. And of course, the higher the concentration of the fructose 2,6-bisphosphate molecule, the better it is for the activity of the PFK enzyme. It can overcome even higher concentrations of ATP being present. But how is this molecule synthesized? So the fructose 2,6-bisphosphate is synthesized and degraded by a bifunctional enzyme that includes both catalytic domains. It seems a little bit counterintuitive that the same enzyme or enzyme complex, if you will, that creates the fructose 2,6-bisphosphate will also destroy it. But this, of course, allows the control by the hormones to happen. So it has a PFK2 domain or phosphofruktokinase 2 domain on this dimer enzyme, which catalyzes the reaction of fructose 6-phosphate plus ATP to form our fructose 2,6-bisphosphate, and ADP is released. But it also contains a bisphosphatase domain, so fructose bisphosphatase 2 or FBPase 2, and that catalyzes, this domain will catalyze the dephosphorylation of fructose 2,6-bisphosphate with an hydrolysis reaction. So how does this dimer decide whether it is going to enact the kinase reaction or the phosphatase reaction, or is this also just a futile cycle of reactions? Well, what actually happens is that adjacent to the PFK2 domain, in each copy of the enzyme, there is a regulatory site which is subject to phosphorylation by that protein kinase A, the cAMP dependent kinase, which gets switched on when glucagon is released. So this protein then can be phosphorylated or it can be dephosphorylated. And which catalytic domain of the enzyme is then active depends on whether that domain is phosphorylated or not. So it doesn't at the same time act as a kinase or a phosphatase. It'll act as a kinase or a phosphatase depending on this phosphorylation. So when the enzyme is dephosphorylated, and that's what we indicate here, enzyme with an OH group dephosphorylated as opposed to enzyme with a phosphate group, the phosphorylated form. So when the enzyme is dephosphorylated, it will act as the kinase and take fructose 6-phosphate and produce fructose 2,6-bisphosphate. But when it is phosphorylated, it will actually act, the kinase activity will be inactive and the phosphatase activity will be active, and this will allow the enzyme to then take fructose 2,6-bisphosphate, dephosphorylate it, and form fructose 6-phosphate. So it is a CAMP dependent phosphorylation of the bifunctional enzyme that activates the FBPase, the phosphatase domain, and inhibits at the same time the kinase domain. And the concentration of fructose 2,6-bisphosphatase will decrease in liver cells in response to a CAMP signal activated by glucagon when blood glucose is low. So glucagon is released, that starts this whole kinase signaling network, which turns on, which we call the CAMP signal cascade, which turns on the kinase A. That'll lead to phosphorylation of this enzyme, and when it is phosphorylated, it will act as a phosphatase and not a kinase, and thereby decrease the amount of fructose 2,6-bisphosphatase present. So when that happens, glycolysis slows down because there is now no more or much less fructose 2,6-bisphosphate to activate the PFK enzyme, and gluconeogenesis will increase because of the decreased concentration of the fructose 2,6-bisphosphate, which would otherwise inhibit gluconeogenesis enzyme fructose 1,6-bisphosphatase. So at that all-important phosphofruktokinase step, this has a converse effect. Either if the fructose 2,6-bisphosphate is present, it'll help the PFK enzyme, as we've seen, switch on glycolysis, even if there's a lot of ATP present. But if it is not present, it cannot, and at the same time, if fructose 2,6-bisphosphate is present, it inhibits the fructose 1,6-bisphosphatase gluconeogenesis reaction. But when it isn't present, it cannot activate the PFK, and it cannot inhibit the fructose 1,6-bisphosphatase. So this is an image that takes together most of the control of glycolysis versus gluconeogenesis. Doesn't have all of the details on, but I will mention it as we go on. So this is the principal regulatory mechanisms of glycolysis gluconeogenesis. Just to take note that the allosteric activators are indicated by a little plus sign, whereas a minus indicates the inhibitors. So what we see is that the glucose 6-phosphatase, which of course is the enzyme which is the bypass of the hexokinase during gluconeogenesis, that's under substrate level control and not allosteric control. So it'll depend on the concentration of glucose 6-phosphate and glucose whether or not this reaction runs in that forward direction to produce glucose. The reverse reaction, of course, regulated by hexokinase, is under substrate inhibition, under product inhibition control. So in other words, glucose 6-phosphate inhibits hexokinase. The fructose 1,6-bisphosphatase is inhibited by AMP and also activated by citrate. That's not indicated here, which is the exactly reverse of glycolysis. So glycolysis would have been inhibited by ATP, activated by AMP, and inhibited by citrate. So what we've also just discussed is that fructose 2,6-bisphosphate activates this phosphofruktokinase enzyme and actually inhibits the fructose 1,6-bisphosphatase enzyme. And the most important allosteric control here is through this reciprocal control of AMP, where AMP activates the phosphofruktokinase reaction and drives glycolysis forward, and at the same time it inhibits the fructose 1,6-bisphosphatase to stop gluconeogenesis from happening. So this just reiterates that fructose 2,6-bisphosphatase is an allosteric activator of the PFK and inhibitor of the 1,6-bisphosphatase, the bypass reaction. And then finally, the protein kinase. We've said it's a substrate for CAMP-dependent phosphorylation, which would help to switch off the activity of the kinase and thereby switch off glycolysis to some degree. It is inhibited by high concentrations of alanine, ADP, and acetyl coenzyme A. Conversely, which is again type of reciprocal control where the kinase is negatively or is inhibited by acetyl coenzyme A, and one of the bypass reactions of gluconeogenesis catalyzed by pyruvate carboxylase is actually activated by acetyl coenzyme A. So then just finally to just summarize the cascade of reactions that happens when blood glucose is low, just to give you a global idea of how this global control works. So when you have low blood glucose concentration, the body secretes glucagon. That increases the amount of cAMP, cyclical AMP, in the cell, which increases enzyme phosphorylation through this cascade of kinase activities that gets activated. That'll allow for the activation of the FBPase2 activity and the inactivation of the PFK2 activity of that difunctional enzyme, which either makes or destroys the fructose 2,6-bisphosphate. So when the phosphatase activity is increased because of and the kinase activity decreased because of the phosphorylation, that'll decrease the overall amount of fructose 2,6-bisphosphate. And when that happens, you have inhibition of phosphofruktokinase because this metabolite is no longer there to activate it, and you have activation of the FBPase because this metabolite is no longer there to inhibit it. And what happens is that glycolysis is shut down, and this increases gluconeogenesis. Okay, I'm going to end this lecture there. Our next lecture will be on glycogen, its synthesis, its breakdown, and the regulation of those processes.